categories were Kapophycus, Euchema species complex, crustose coral and algae, Tripneustes compressa, reddish brown crust, limestone, other algae, and other coral. Limestone, it covers both limestone and turf algae. I initially split these, but based on how people were recording them in the field, decided to lump them together for the analysis. So what did we find? At the start of the study, urchins remained clustered around the reef location. So these are frequency histogram bars, and right here is next to the reef stake. And then you can see as over time, density decreased as urchin dispersed. The numbers in red are the numbers of urchins found. The number of urchins found, unsurprisingly, decreased over time. They became harder to find and monitor. And there's really two main reasons for this. As collector urchins, they collect things. And so as you are farther away from the release point, they become harder to find. And although we became very good at spotting urchins in the field, I'm sure some remained undetected, especially as they were farther from the release point. Also, and this was a bit of a surprise, they went down the slopes and kept going down the slope. And so you can see here, there's an urchin, and it's in the middle of a Tripneustes compressa reef area. And as they went further down, we were snorkeling and just were not able to find them. So they had a very slow dispersal from the release point. At 4 weeks liberty, so 4 weeks after release, 90% were less than 10 meters from the release point, and at 12 weeks, about 76% were less than 10 meters from the release point, indicating that a large portion of the Tripneustes seeded on a reef for biological control purposes will be expected to remain close to the release point. Although 30 urchins were surveyed the last observation day, only one of those was tagged. The rest of the urchins were untagged. The reef was surveyed extensively prior to the study, and there were no urchins on the reef beforehand. This indicates that all of those untagged urchins were actually our experimental urchins and had lost their tags. Tag loss actually started only 7 days after release. For the last survey date, 97% had lost their tags. So tag half-life was about 40 days. You can see here that it was as the screws they just unscrewed from the test. There's a small necrotic area, and this closed up in a week. In a week, you actually couldn't tell if the urchins had ever been tagged or not. We didn't observe any mortality or an increase in the necrotic area. So why was there such a difference between this study and the pilot study? And I don't have a really clear answer for that. Bay 34 is definitely more exposed in the northern portion of the bay. It has more crevices, and urchins definitely went into the crevices, and that could have dislodged it also. Kapophycus being that really rigid morphology, maybe by covering themselves with that, it dislodged tags easier. This actually did affect researcher ability to track and monitor the urchins because preference and movement teams worked independently. We combined the data later by unique tag numbers. So a lot of the analysis that I'm going to talk about next is just with that first month of tagging data. Afterwards, there's just so many untagged urchins that, in terms of movement particularly, they couldn't have been tracked. So these tags are not recommended for long-term tracking using this procedure. So the estimated daily net displacement, that was the pink line on the triangulation primer. Almost 40% were under 20 centimeters, and I actually consider this in the realm of surveyor error in terms of the urchin might not have moved, but it looked like it moved a very little bit. So if we increase that to 40 centimeters, still not very far, 64% were under that. There were observations over 100 centimeters though, but this was only when the time between observations was 1 day. There's actually two over 500 centimeters as well. As the time between observations varied, one of the 2 dates that we observed consecutively on sequential days was analyzed further to elucidate daily net movement. So I looked at the movement from March 7th to 8th with 46 urchins. The range was between 5 centimeters and over 500 centimeters. This gives us an average of about a meter a day, but a median of only about half that. This indicates that urchins probably move in spurts. So they're taking a burst of movement, and we're only capturing that if we look at them every day. If we look at them over a longer period of time, like most of the observations, that's just as it gets divided by more days, you're just not seeing that spurt of movement. Over a large time scale, urchin movement looks like it actually may be directional. So they appear, at least some of the urchins, to be moving away from and returning to the same or similar location. This was on the reef flat. So to navigate ourselves around this is distance along the reef crest, and the y is distance looking from above. So this middle line right here is about the reef crest. This is the slope, and this is the flat. So in the flat area, you can see some urchins appear to be going back to the same area. This urchin actually made a loop in about 2 weeks and returned to the same area. You can see that with a couple of the other urchins as well. The ones that went down the slope, however, were unidirectional. Once they started to go down the slope, they didn't turn back ever. On this graph, the blue bars represent benthic frequencies based on quadrats. So these were the frequencies that we would expect that urchins would be eating if they were indiscriminate feeders in the field. And the red bars were the frequencies that urchins actually ate, what there was actually underneath each urchin. A chi-square goodness of fit test indicated that there were significant differences between the actual and expected frequencies. Urchins were on top of crustose, coral, and algae much more than expected, and actually on Capophycus and Prides less than expected. Feeding preferences appear to be related to urchin size. So just looking at the Capophycus blue bars here in this graph, you can see that as urchin size increases, the time that they're spending on Capophycus is decreasing. However, the number of urchins in the smallest and largest size classes is really small. So even if you ignore those columns, and even just look at the two columns that I have the most number of urchins for, you're still seeing a decrease between about 8 and 9 centimeters diameter. The last portion of the results section is talking about the comparison of the substrate under the urchin to what was actually in the Aristotle's lantern when we turned that urchin over. So we're looking at what we observed to be under them versus what is actually in this apparatus. This was just done on the last 2 sampling dates. So our categories for this were Capophycus, crustose, coral, and algae, limestone, reddish-brown crust, and Prides compressa. So just looking at what's underneath the urchin, there we had 65 individuals. So these are not going to add up to 100, they're going to add up to 65. So most of them, what was underneath that we found was crustose, coral, and algae. When we turned those over to see what they were actually feeding on, so 14 here means out of the 16 that were observed on top of Capophycus, 14 were observed eating Capophycus. And to look at this a little bit more clearly, this percentage indicates the number that are feeding on what they are on top of. So in this case, 50% of the urchins on top of crustose, coral, and algae are actually eating crustose, coral, and algae. You can see a number of instances here because most of these don't add to 100. So if they're not feeding on the substrate that's underneath them, is there anything actually in their mouth? So crustose, coral, and algae, for instance, Capophycus was found in that Aristotle's lantern 6 times out of the 34. So they're actually carrying that with them to an area outside of Capophycus, and they're still eating it. To summarize this, when they're on Capophycus, they tend to be eating Capophycus. and they're on crust